Hello. Thank you, Jenny and Cliff, for that wonderful, very sophisticated talk. I'm now going to go to the world of babies, which gets a little noisy and perhaps a little less sophisticated, but let's see how we do today. So what I'd like to talk about clearly here is disparity-driven reflex vergence in infancy and early childhood. And I'd like to talk about the clinical implications of all of that. I am an optometrist by training, but I have deviated from the clinical, practical, everyday path and have been thinking more in the research lines for a number of years. So when I went to optometry school, Hubel and Wiesel had done their fantastic Nobel Prize winning work, and we were starting to learn about the receptive fields, however Jake would like to define them. And we'd started learning about the impact of abnormal early visual experience and how that changes the ocular dominance patterns as one metric in visual cortex. And so as a new young optometry student, I was very excited about this. And I assumed that next week I'd go back into the next lecture and be told how to act as a clinician and optimize the visual experience of young infants and prevent all of these problems. And here we are 30 years later, and I'm still thinking about that. So what I'd like to talk about today in the context of functional vision is really the notion of trying to sort of prevent some of these atypical developmental paths that some of our children with particularly binocular vision disorders today, but I like to think about this more broadly in terms of children with visual impairment as well. The work of Hubel and Wiesel and others looked at the impact of abnormal afferent activity coming into the brain on the visual cortical development. What I, running around thinking about the typical human infant, started thinking about was, wait a minute, we have the motor piece of the puzzle. How is the young infant controlling its motor behavior in order to generate the appropriate sensory input? And so this visual motor loop was actually what's defining the visual experience of the young infant, and that may be causing atypical development of cortical structures. So if we think about that question, we can start to think about what was known at the time when I was a young student. As I think back of 60 years of Smith-Kettlewell, this set me off down this path, and now you're suffering listening through it, I'm afraid. So what did we know when I was a young optometry student? Well, Davida Teller had been working on developing techniques to assess the sensory capabilities of the young infant, and she and her husband Tony here worked together to develop a simulation of what the vision of young infants might look like. We have the newborn, the 1-month-old, the 3-month-old, and so there's limited contrast, sensitivity, limited color in the vision of the young infant. And as I start to talk about disparity-driven vergence and binocular function, at the time, there's very much the notion, courtesy of a number of studies, that while visual acuity develops slowly over a number of months, the development of stereopsis, or the ability to sense disparity, was developing very rapidly at somewhere between about 3 to 5 months of age. And so there's this rapid onset of binocularity at this point in time. Well, there I was sort of starting to think about, well, what's the experience of the infant and how's the visual and motor piece working? This set me off down a path to try to understand how binocularity could develop with these early capabilities of the young infant. So today what I'm going to talk about is just a couple of studies we've been doing in the lab. It's a smaller part of sort of the larger program, but it's specific to the disparity-driven vergence piece. So one of the first things we can do is say, okay, the real world is actually very dynamic. It's not a random stereogram and taking some time to figure out Chris's tricky magic eyes. We actually have to do things on the move as a young infant. So if we put a screen mounted on a track with a little beam splitter, we could now maybe move this screen back and forward and then capture the motor responses of a young infant while they are looking through a window here that holds their head within the viewing aperture of this camera. What we can do, courtesy of some nice new continuous psychophysics. It has been used for primarily, but continuous tracking protocols. What we can do courtesy of those protocols now is move the screen back and forth on the track randomly, as shown in the video at the top there. And we can start to look at how the infant now, as this screen is moving back and forward, how their accommodation and vergence are responding to this dynamic stimulus. So what's shown here is eccentric photorefraction, the pupil looking bright. We can use the distribution of light in the reflected image there to assess where the eye is focused at the moment in time. We can also do the Purkinje image eye tracking to estimate eye position. So if we start to take very young infants and we present them with these randomly moving targets with the screen on the track, how well do they track them? What's the sensitivity of the vergence system? How well is the motor system functioning in this early period after birth, where the binocular system should be coming online? If we look here, this is one track or one profile of the stimulus motion over time. The stimulus is moving randomly back and forth on a screen in front of the infant, and over about 100 seconds takes on a very random profile over time. If we look at data from individual subjects here, what I've plotted here is the accommodation from the right and the left eye and the vergence response from the two eyes together for a single adult and an 8-week-old infant. And what can be seen here is that the adult tracks with their vergence eye alignment quite well, the structure of this stimulus over time, and their accommodation system also does the same thing. Totally unpredicted in my mind, given David Teller's simulations of immature vision of infants in the first months. At 8 weeks, this single trial from an individual 8-week-old is also tracking this random motion of the screen on the track quite well, both in terms of vergence and accommodation. To quantify these responses and give us a sense of the development of these processes, if I take the stimulus plot and take the response of either the accommodation system or the vergence system, we can correlate the stimulus to the response and see how well the two of them correlate with each other, but then also shift the relationship between the stimulus and the response in time and do a cross-correlation to see the time at which we get the best correlation between the stimulus and the response. If we do that, what we find is that the adults have a best correlation, a peak correlation on the order of 100 milliseconds, 200 milliseconds, something like that, for both the accommodation and the vergence responses. And this single 8-week-old infant has actually a similar correlation, but on a slightly different latency. And this is all the PhD work of Colin Downey shown at the bottom here. He tested a lot of infants, and so let's see what these correlations and latencies look like during this early period of development. Here are the data for vergence. On the x-axis, we have age in days now, not in months, and on the y-axis, we have the latency of the vergence response. Each individual point in this graph is a single 100-second trial, and on the color is an indication of the correlation strength between the stimulus and the response. What you'll notice is that while at the youngest age in days, 25 to 40 days, the correlations are quite low between the stimulus and the response, by something like 2 months, we have got almost adult-like correlations in terms of color, and the latencies are coming down. And so by a couple of months of age, these young infants, in terms of their accommodation and slightly more noisily, but heading there, in terms of the vergence is quite good. The accommodation is now a little noisy, but is on its way also. This was really surprising to me. I thought we were going to be writing NIH grants and tracking the development of these responses for years, and there'd be a whole profile. But what we could demonstrate here is that these motor responses defining the visual experience coming into the young infant are maturing quite rapidly, and by on the order of 2 months or so, the coordination is there to get nicely focused retinal images and nicely aligned retinal images. And I can speak to that again later, but this has implications clearly for those of us thinking about screening of young infants and looking out for them trying to predict atypical development. Okay, so if the responses are that mature, what's driving them? As I said earlier, we'd known for a number of studies that infants start to respond to relative disparity in stimuli at about 3 months, 3 to 5 months of age. What's driving a vergence response within a couple of 100 milliseconds to an almost adult-like accuracy at a month and a half, at 6 weeks or so? Well, first of all, we could say, how well are these infants actually doing it? Could the younger infants do better? These are 100-second trials. We know infants aren't terribly alert. What's going on there? So here, what we did in this analysis is we did this cross-correlation just for limited periods of time, just for 10 seconds, all the way through the 100-second trial. And what we can then do if we do these limited cross-correlations is we can think about what's the correlation during the course of the 100-second trial. And we can look at whether behavior waxes and wanes with infant attention and alertness. If we do that, in other words, plot the distribution of correlations on the y-axis here in this plot, we can look for an individual trial now. Each point on the x-axis is an individual trial. We can look at the distribution of correlations in that 100-second trial for each of these trials all the way through as a function of age. And so we've got the 5-week-olds, the 9-week-olds, 13-week-olds sort of marked in age there. And the adults to the right-hand side here, we can see if we look at the distribution and the probability of all of these correlations, we can look at the fact that by 9 weeks or so, we're seeing distributions of correlations that might be a bit variable, but they are actually heading up towards the adult value. They are tightening. And so we're seeing a lot of maturation early on, before the sort of 3 to 5 months where we really thought we should be concerned about binocular function. Here are the same data for accommodation. They're a little more noisy, but they generally show the same trend. Okay, so back to my question of what's driving this apparently immature, unexpected performance. Then one question to ask there, sort of a bold question, if we ignore that previous literature and think about the motor system, can retinal disparity actually drive vergence as opposed to relative disparity responses in the context of stereo and maybe perception? Can retinal disparity drive vergence as an oculomotor response early on, prior to that previous literature looking at the relative disparity responses? So what we did to look at that question was to take a random field of large checks, because infants have immature sensory responses to spatial frequency and contrast, take these large checks, change them every 1/3 of a second, so that now we have no structure in the image to pursue, to make saccadic eye movements to track. We just have an updating noise field. And now we can set it up so that the only thing that changes over time is disparity of that noise field in the 2 eyes. We have exhibit A, the young infant, being held by exhibit me, and we can see what we can get using our eye tracker here. What you can see now is the stimulus just going sinusoidally over time at a frequency of 0.1 hertz. And if we look at the responses of the 4 youngest infants in the study, there is structure tracking the stimulus in individual responses at those youngest ages. And so in summary, this graph here plots age in days again on the x-axis, and we've just plotted individual infants at their appropriate age in days. And then longitudinally, there are colored symbols that indicate repeated measurements from single infants as they aged. What we can see here is that the response at the temporal frequency of the stimulus is much greater than the local noise estimate at adjacent frequencies for a good number of these infants. And in fact, that a number of them can generate disparity-driven responses that are close to the amplitude of the stimulus. Not all of them are responding to disparity, but a good number of them are by 2 months of age. And so this combination of this couple of studies really emphasizes the fact that the motor system is functioning far more maturely than certainly I would have anticipated as I went into this 20 years ago or so. So that's a start. As I think about clinical care, now we want to start to think about differences between images. So the next question that I just want to spend the last few minutes on here is thinking about how much anisometropia does it take to disrupt reflex vergence. And so as Jenny Reed was beautifully laying out in the previous talk, disparity will drive a reflex vergence movement. But if we now start to disrupt the relationship between the images in the 2 eyes, how easy is it to disrupt that vergence and therefore to start to think about atypical development in young infants and amblyopia and strabismus? So that's the last question I want to think about here. The relationship between amblyopia and anisometropia is quite complex. And if anyone is interested in that, Brendan Barrett and Arthur and I have had a review in PRER in 2013, where we laid out how confused we were about the topic, at least. So what we need to do to think about how much anisometropia is too much, we need to present the stimuli dichoptically. So we took a ProPix projector with an active polarizer. We then had a large rear projection screen that maintained the polarization. And we, on that screen, produced small little fun targets, 2 degrees by 3 degrees, just centrally in the screen there. And then we put our iLink eye tracker in front of that with a beam splitter and circular polarizing glasses. So that's the setup. And then we have Exhibit C, the slightly older child there, who's sitting looking at these small images that they're being presented with. The next part of the puzzle was to say, okay, now we need to introduce our defocus. Well, we could think about it in very straightforward, let's go to Photoshop and introduce Gaussian blur, sorts of blurring, but we wanted to do this accurately. It's an important part of the puzzle. We've got to figure out literally how much blur in an eye is too much blur. And so to do that, we actually simulated spherical defocus in the retinal image. We measured the pupil size of each participant at the beginning of the study and then used a convolution of the point spread function that was calculated for their pupil size, convolved it with those images to produce accurately defocused images to present in this project. We presented the blur both dichoptically to both eyes and to one eye to mimic anisometropia. We collected data from adults, typical boring adults, and then looked at their tracking responses in a continuous, now same idea, continuous tracking, stimulus moving back and forward in disparity, back and forward in depth, looked at their ability to track that stimulus. So now to look at bilateral defocus to both eyes and then right eye defocus, left eye defocus, what do we get? If we look at the peak correlation of the response to tracking this motion in depth, we can, for these adults, there was very little effect of defocus here on the x-axis, from 0 defocus to 4 diopters of defocus in both eyes. There was little effect of the defocus there. But now, as we start to blur one eye or the other eye, we start to see vergence being impacted within about 1 to 2 diopters of defocus. Interestingly, if we then went and did the same cross-correlogram, looking at the relationship, the correlation between the gaze of the left eye, the gaze of the right eye with the stimulus, we can look at the impact on the motor behavior of the two eyes separately in this dichoptic task. And what we can see is when the right eye is blurred, the right eye's tracking ability is a lot more impacted than the left eye's tracking ability. And that flips to the left eye being more impacted when the left eye is blurred. And so there's a clear asymmetry there, which I hadn't necessarily anticipated ahead of time. But that was quite a dramatic result that we could see in the adults. So let's see what the children did. Sorry, forgot one part. As performance decreases with the correlation here, we can start to say, well, what are the eyes doing then if they're not tracking the target well? So to look at that question, we started to say, okay, well, if it's not vergence tracking, is it version? Are the eyes now moving in a conjugate sense as the vergence performance starts to fall apart? And what we can see is, yes, as vergence started to be affected, we started to see more correlation in the version movement, as though the focused eye is now taking over control of the both eyes in a conjugate sense. What did the children do? So we took 22 children from 4 to 7 years of age, and now we could see again the reduction in vergence performance in correlation with the increasing defocus. And once again, we could see with the children the impact of the defocus, increasing defocus on the eye that was defocused, that flipped as we switched the defocus to the other eye in the case of anisometropia. And a hint that perhaps the version correlation is increasing a little bit there too. So in summary, to wrap up, disparity-driven vergence responses can be generated in the first 2 months after birth. And the vergence characteristics are quite mature in that continuous tracking profile and for dynamic stimulus by about 3 months of age. This is much younger and better performance than certainly I was thinking about when 20 years ago. This has really highlighted the fact that we shouldn't be waiting till 6 months of age, as I've heard some clinicians say. We shouldn't be waiting months and months to make sure that an infant has typical visual experience. Simulated unilateral defocus has more impact than bilateral defocus. That's consistent with some previous results in terms of stereopsis and perceptual performance. But now we can really see the impact of this in the motor system. The intraocular differences in peak correlation with unilateral defocus do reveal the intrusion of conjugate version responses driven by the focused eye. And so this all is starting to, I hope by the time I finish my career, thinking about this first when I was starting my career, I hope that we might be able to start to think about some screening tools to have, with all the new technology, perhaps that we've been talking about this morning, to start to think about being able to do a simple screening test to look for prediction of atypical behavior much earlier than has happened in the past. And I will stop there.
Thank you, Rowan, for a lovely talk. Excellent. We've got time for a couple of quick questions, 1 or 2. Preeti?
So Rowan, I know you want to use it as a screening tool, but I'm sure you've probably already gotten some data from actual anisometropes. Do they look like that as well?
We're just starting to do that. So this was literally VSS in May. So COVID put a damp on our progress. Yes, so that's absolutely happening. I don't have anything coherent enough to give you a decent answer, but yes, we've got a little bit of data. We're just getting that going. Absolutely. Yeah. And high hyperopes, people at risk for strabismus. We're trying to look at all of it as a potential tool.
Thank you. Any other questions? Yes, Ben.
Thank you. Well, I think I mean, it looks very compelling that we ought to be screening infants for unilateral hyperopia very early and getting in the glasses really early. I wonder if you have any thoughts about the politics of how to make that happen in this country and around the world.
Especially here.
So this is another favorite topic of mine, and it's really a pub topic with beer. I could talk extensively about this topic. So if we think about 1 to 2 diopters of anisometropia in early infancy, we're actually talking about the period of emmetropization. And so refractive error tends to diminish over the first months after birth. So actually, as we get into the subtleties of this, there's this trade-off between permitting or trying to encourage emmetropization, will they lose the anisometropia, and protecting the development of the neural system in order to allow it to function. So there's a subtlety there that I only really spoke about disparity-driven vergence today, but there's a subtlety there to the whole picture. And we have ophthalmologists in the room, Suzanne, who can speak to this because they've experienced it and have a lot to say about this, I hope. And so there's some subtlety to how it might be managed in the very early period of time. We've also got the question, I haven't demonstrated that this atypical motor behavior is enough to disrupt long-term development. Right? So it's feasible that we might see a bit of disruption here, but if the anisometropia went away, then maybe everything's fine. And so I'm digging myself a big hole here because I'm just making it way more complicated. But I think in order to venture into the sort of political discussion that we're talking about here, we've got to have really solid ground because there are a lot of people with strong opinions about all of this. And I think I want to work really hard for the next few years to try to get a solid footing on which to say, here's the evidence. Let's do this, whatever the evidence ends up being. But we need the evidence to really make a strong argument. And then I hope everyone will come to the table.
Thank you. I think we should hear some of those strong opinions in the general discussion, but in the interest of time, Iwa is going to.
We have a question in the chat.
Oh, there's a question in the chat.
So I'll go ahead and I can go ahead and read it out. So the question is, please share your thoughts on assessing and treating problems in BOV in children with multiple disabilities.
Can we save that to the
Yeah, let's save that for the
discussion.
the general discussion.
Okay. Yeah.